Let's begin with comparing a plant found in a desert to a plant found in a tropical rainforest. Although their phenotypes are similar, the genotypes differ due to the transcriptional mechanisms. Both have a waxy coating, but the genes in the desert plant code for the wax to preserve moisture, while the tropical plant repels moisture. This type of gene expression altered by the environment can be applied to the case study with B. antarctica insects. Before we begin, here are some key terms you might want to know. Arthropods are invertebrate animals that have a segmented body, joint limbs, and an exoskeleton made of chitin. Desiccation and chiroprotective dehydration are two different processes of induced drought. Upregulation is the increase of a cell's component, and downregulation is the decrease of a component.
Arthropods are very prone to dehydration due to their small body and high surface area to volume ratio. In this study, B. antarctica, the southernmost insect in the world, will be studied. They can survive up to 70% water loss, making them extremely tolerable of dehydration. Researchers used RNA sequencing in order to locate differences in the transcriptional mechanism. Changes in gene expression in response to dehydration indicate that cells increase their cellular recycling pathways like autophagy and decrease their production of ATP. The results also indicated that there is a strong correlation between gene expression and modification of general metabolism. Additionally, the gene expression of B. antarctica was compared to that of M. arctica. The results of this comparison show that there is little overlap in gene expressions of both arthropods, even though they live in similar environments. Therefore, it is assumed that transcriptional changes were mostly species-specific. This indicates that these arthropods have developed distinct transcriptional mechanisms in order to adapt to extreme conditions.
This experiment was conducted by Nicholas M. Tietz of The Ohio State University. Its purpose was to show how metabolic processes are affected by dehydration. The alternative hypothesis was that the gene transcription changes are specific for individual Arctic arthropod species, but have the same effect in helping to prevent dehydration.
As stated before, the arthropod being tested was the species B. antarctica. The three trial groups consisted of the control group, desiccation group, and cryoprotective dehydration group. Each group was kept at the temperature and percent humidity listed. The arthropods lived under these conditions for 5 to 10 days. The desiccation and cryoprotective dehydration conditions caused them to dehydrate by 40% of their water levels. At the end of the time trials, RNA sequencing was used to measure the genes' expression levels in each group.
Before looking at the results, here's some more vocabulary terms you should be familiar with. In this case study, autophagy is a process that recycles cell components to conserve energy and prevent cell death. Heat shock proteins, also referred to as HSP, repair misfolded proteins caused by dehydration. HSF is the enzyme that regulates HSP expression in the cell. Cestrin is the gene that inhibits cell death. Hexokinase is an enzyme that synthesizes glycogen 6-phosphate, which is the glucose in sugar.
The results of this experiment found that, in general, desiccation and chiroprotective dehydration yielded similar components that were upregulated and downregulated. This, in turn, led to the same processes that were affected. It was found that dehydration promotes autophagy because autophagy reduces cell death by recycling cell components in times of stress. The recycling of cell components of autophagy was increased due to the upregulation of cestrin. The upregulation of signaling and protosomal genes also correlate with the increase in autophagy. The upregulation of the cell component recycling pathway serves the purpose of conserving energy when cells are experiencing dehydration.
In turn, it was found that the downregulation of certain genes related to ATP production and metabolism also benefited the arthropod to survive dehydration. It was noted that nearly every gene involved in glycolysis, the citric acid cycle, and ATP synthesis were downregulated. This downregulation helped contribute to the energy conservation. After the experiment concluded, the scientists compared the results with previous case studies. They found that arthropods in different environments have different gene transcription responses to dehydration, but always result in the same energy conservation processes. Although the two dehydration processes showed similar up and down regulation responses, the gene expressions tended to differ. While around 1,900 genes were expressed similarly, 1,400 genes in desiccation and 450 in cryoprotective dehydration were expressed differently from the control group. This Venn diagram shows the differences in genes that were not similarly up or down regulated between the two. In comparison, 37 genes were regulated in desiccation differently than the 2 upregulated genes in cryoprotective dehydration.
So how does this relate to our class, you may ask? Well, this experiment showed how the differences in gene expressions can be caused by the environment that the organism lives in due to adaptations. It also showed how the regulation of enzymes and processes caused by glycolysis and the citric acid cycle to stop so that the ATP input would not be wasted. This allowed for the cells to conserve energy and therefore survive the harsh conditions that they were placed under.